Well, Mike, before we get started, I'll just sort of talk to everyone about a little bit about the Science and Culture Network. So the Science and Culture Network, there are basically three chapters of the Science and Culture Network. We're the Southern California chapter. We also have a chapter that's in Denver, and there's also a chapter in Houston. So if you happen to be in any of those cities or in those surroundings, then please do reach out to those, and we'll provide you some more information at the end of this. We are recording this, and we will try to provide this to you if you would like to show it to others, because this is a public forum. So basically, the Science and Culture Network is we basically are affiliated with the Discovery Institute, and as you guys probably know, the Discovery Institute is a proponent, amongst other things, is a proponent of intelligent design. And so what we've done as being a chapter of the Science and Culture Network, affiliated with Discovery Institute, we have the great opportunity to interact with some of the great minds in intelligent design, Michael Behe being one of the stalwarts of intelligent design. And we thought that this would be an appropriate time, and that it would be really enjoyable for people to have a conversation, or at least to hear Michael and Dr. Behe, and then have that opportunity to ask him some questions in terms of his views on viruses, particularly COVID-19 and evolution. So I will stop there, and I will turn it over to Dr. Behe. Dr. Michael Behe, thank you so much for joining us today.
Thanks, Walter. It's great to be with everybody. I was contacted by the Southern California chapter of the Science and Culture Network, and they were interested because, of course, of the pandemic and talk about the evolution of viruses and so on, to present a talk on those topics, on viruses, COVID, and evolution. So this is going to be a broad talk, and I would like to start with a little bit of history. Here we go. And as you know, disease and suffering and so on, did not start a couple months ago. It's been going on for a very long time. Plagues have been around for pretty much all of recorded history. And on the screen here is a painting by a later French artist of what he imagines the scene looked like for what's called the Antonine Plague, which took place around 165 AD or so, during the reign of Marcus Aurelius, the Roman emperor. And you can see that all kinds of people are just keeling over in the streets, dying. And if you can see up in the corner, here's a funeral procession coming down the street. And here the artist puts in the angel of death knocking on the door. And there is, of course, some artistic license. But as far as the citizens of Rome at that time knew, heck, it may just as well have been some, fantastic supernatural agent or something. They had no means to understand what was going on. They just saw people dying, and then more people dying, then most people dying. And of course, many plagues occurred after this one as well, but it was only about, it took about 1,500 years, 1,500 years before science started to understand what was going on. The problem is, of course, that we humans have wonderful senses, but they do have their limits, and we can only see things that are so small. And yet we have now learned that the foundation of life occurs at a level that's too small to see with the naked eye. And so we had to wait for the advance of science, the development of new tools, in order to even begin to understand what was going on. And in the second half of the 17th century, the microscope was first introduced, for use in science. And as soon as it was used, people were astounded because they saw these tiny little creatures that they were familiar with, like insects, and they saw that they had innumerable details and weird features that no one had ever suspected. For example, the compound eyes of insects. It looks like some sort of space alien. People had never seen that before. They saw that some fleas had tiny mites on them, so that it seemed that this one could shrink the living world continuously. And as a matter of fact, that was the occasion for a little ditty that was written by this guy here, Jonathan Swift. You probably know him as the author of Gulliver's Travels. He wrote, "So naturalists observe a flea has smaller fleas that on him prey, and these have smaller still to bite 'em, and so proceed ad infinitum." So he was thinking that, well, that one flea, such as in the last slide, had those smaller fleas on them, or smaller insects. And he imagined that that one would have a smaller one, and that one, and that one, and that one. And it would go on forever. And it's hard to appreciate it from our time period, but this shook the foundations of people's understanding of the world. It turns out there was an invisible layer of life that nobody had even thought of before. There's a historian named Charles Singer who wrote about the history of biology a while back. And he says of that time that the infinite complexity of living things thus revealed was as philosophically disturbing as the ordered majesty of the astronomical world, which Galileo had unveiled to the previous generation. So in both cases, looking at the stars, it seemed that space and the universe was a lot bigger than people had thought. And looking through the microscope at life, it seems that life was much, much, much more detailed than people realized. And since that time, we have not stood still. Turns out that science can advance even more as that new technology is improved with better technology. So the first microscopes were pretty crude, but then better microscopes were invented that had better lenses and stronger lighting, electric lighting, and stains and oil immersion and all sorts of things that could ramp up the resolution of the microscope. And then a short time after World War II, the first electron microscope was invented, and that increased the resolving power even further. And even now, we are seeing the complexity of life in ways that we find philosophically disturbing. For example, just a few years ago, in the year 2013, some scientists published a paper that used a new videographic technique, a new way of making movies. And they examined this rather common insect called a plant hopper. And they looked at the top of its legs because they wanted to know how it could jump so far and so fast. And they saw these little bumps, and they used their new technique to look even closer at those. And they saw that those bumps were gears, literal mechanical interdigitating gears. And this was astounded pretty much everybody who read about it. And the internet lit up for weeks with philosophical and even theological discussion of the implications of finding gears in insects. So the point is that back in the day with Romans, people could see the anatomy of living creatures. And there were a lot of smart people back then, even scientists. There was a physician named Galen, and Galen dissected animals and looked at the anatomy of various organisms. And he could see down to about this level. I don't know if you can see it on the screen, but there's a little circle here. And this is blown up to this. And this is a piece of skin. This little circle blows up a piece of skin on this fellow here. And this is me when I was a postdoc about 40 years ago. And then you can see this little region here is blown up about 30-fold to show this. This is a piece of tissue, the epidermis. And then another 30-fold, just this little region to blow up a cell. This is a basal cell, it's called. And then this region shows the mitochondria. And this region here shows the piece of the membrane of the mitochondrion. And you get all the way down to here. And this is a protein molecule. And as we've learned over the past, decades and century, atoms and molecules are the foundational level of life. And this is a single molecule composed of hundreds of atoms, but it's a single molecule. Now, Galen and the folks in the Roman days could see down to, at best, around this level. And with the advancement of microscopes, we were able to see down to about this level. But in order to get further, it took the electron microscope and also different techniques to see what proteins looked like. And it turns out that the problem that the Romans had in the Justinian plague was caused by biological agents whose size was around the size between here and here. Viruses and bacteria have sizes that are about 100 million times smaller than a full-fledged organism. And so we had to wait for the development of techniques that allowed us to see those before we could understand what was causing these plagues. This thing here, this is a mitochondrion. This is roughly the size of a bacterium. Now, a virus is about 1/10 of the size of this. Viruses are generally considered to be non-living, whereas cells like bacteria are considered to be living. So let's take a look at one of the first viruses that was examined, and that's shown here. And this is actually a virus which does not cause human disease. It instead invades bacteria. It will get into a bacteria and kill it. And so the viruses of bacteria are actually called phages, which means eat. Bacteriophage means it eats bacteria. And this thing here is called bacteriophage T4. This is an electron microscope image of one bacteriophage T4 in the process of infecting a bacterial cell. And as you can see, it has some very definite structure. It's got this big head thing and like kind of a long region of stalk, and then this smaller stalk too. And with a lot of investigation of this is pretty fuzzy and so on, but people put together models of what that virus looked like. And here you can see this kind of geometric figure called the head. And this is where the genetic information of the virus is stored. It's stored inside here. It's a piece of DNA, much smaller than human DNA, but enough to code for about 50, 60 different proteins. This is just a stalk, which is composed of other proteins, and it's hollow. And the DNA, when the virus gets on the cell, the DNA from the head is shot down through, whoops, that was a mistake, is shot down through into the bacterium from the bottom of it. Now, let me just emphasize that this is not a cell. This is just a collection of molecules. It's not really a living thing. Most people don't think. It's only kind of a package of genetic instructions. Well, in recent years, in the past 10 years or so, the science of microscopy has gotten even better and has improved resolution by quite a bit. And so people went back and looked at T4 using a technique called cryo-electron microscopy. Cryo-electron microscopy. And a study that was published just a few years ago concentrated on just this region, this tiny region of the whole virus. And this region is called the base plate, And it's shown on the next slide here. This is the cryo-electron microscope image of that. Here's the base plate down here. And you can see the much greater detail. And this region right here is blown up. If you can see the little dots, it's not clear, but if you can see the little dots, each one of those dots is an atom or a group of atoms. And they are so arranged so that they stick to each other in the right order to give this structure. And this is the tubular region, and it again is composed of protein molecules, which have each of which has many atoms, and they're arranged just so that it can form this structure. Now, this here is the structure of the bacteriophage T4 before it attaches to a bacterial cell. And over here is an image of it after it has attached to a bacterial cell. And you can see this is shorter and fatter, the stalk region. And that's because when it touches a bacterial cell, this contracts rapidly and it shoots a tube down through the membrane of the bacterium. And the DNA from the head of the bacteriophage is injected into the bacterial cell. So this is essentially an automated syringe that the bacteriophage uses to get into the cell. And again, this focuses on the arrangement of the proteins in this tiny area. And all these are just atoms which are taking part in the process. And if you can see, the arrangement here is somewhat different than the arrangement here. And that's because all of these things move in a coordinated fashion as the bacteriophage injects its material into the cell. Okay, so let's look at this again. Here again is the bacteriophage. Here again is the base plate. Now, with the advances in computers and microscopy, some scientists have been able even to make models of how mechanically the bacteriophage works. And here's one for bacteriophage T4. This is a movie made by the investigators looking at this. Each of these things, if you see little details in here, they're atoms or little groups of atoms. And all of these blue regions, they're different sorts of proteins. This is made of another kind of protein, and they're all coordinated in their motion. It's like a space lander. It's a real molecular machine. Okay, now I showed you this because we have a lot of information on the virus bacteriophage T4. It's one of the most highly investigated viruses around. But let's take a look now at one that is of more urgent concern to us these days. This is an image of the coronavirus, COVID-19, that is causing all the problems of the last few months. And if you look at this, it turns out that the size of this is roughly the same size as bacteriophage T4. Doesn't have the same structure, but it's roughly the same size. And you can see that it's kind of like a ball that has spikes on it. And like bacteriophage T4, this is just a collection of molecules, of proteins and so on. There's no metabolism inside it. There's no liquid really inside it. These are just proteins stuck on other proteins with fatty molecules called lipids around it as well. And COVID-19 uses a different way to get into the cell. You see that it's not set up like bacteriophage T4 to inject its genetic information into a cell. And in case of COVID-19, the genetic information takes the form of RNA, the chemical cousin of DNA, but pretty much does the same thing. So the next slide shows us the general way in which a virus like COVID-19 gets into a cell. These little blue things that you see on this well, this is the virus, and the blue things are the spikes that are supposed to be sticking out from it. See this little squiggle in here, that's supposed to be the RNA genetic material inside the virus. And the virus comes and one of these spikes interacts and sticks to a receptor molecule. And when it sticks to it, the cell has machinery that thinks that this is a signal for it to pull the material inside the cell. So the cell uses its own machinery to then pull the genetic material of the virus into the cell. Once inside the cell, the genetic instructions that are coded in the RNA of the virus hijack the molecular machinery of the cell and use it to make more copies of the viral RNA, more copies of the protein coats that the DNA will live in, and more copies of the spike protein that will go on the surface of new viruses. And then after a while, all of these things get put together. The virus goes to the surface of the cell and buds off, taking some of the membrane of the cell with it. And now you've got a bunch more copies of the virus where you had only one before. And these can go on then, each of them, to hijack and take over another cell. a different lung cell, say. Now, one thing you have to keep in mind is that these little things are cartoons, and they're vast simplifications of what is necessary here, what is actually going on. When the virus interacts with the receptor, the purpose of the receptor is not just to bind a virus. The receptor doesn't want to bind a virus. It has a job specific to its role in an organism, like say lung tissue or something. So this is a protein that's doing something helpful for the organism. And the virus happens to have a protein that has a similar shape and complementary properties to the receptor so that it can bind strongly to it. How does it do that? Well, let's go to the next slide. So here is kind of an inflated view. These are not the proteins under consideration, but they illustrate the point. Here is an expanded view of what a protein might look like. Each one of these little spheres would be an atom or a group of atoms in a protein. So if you consider this to be the receptor of the cell, and this the spike of the virus, then this guy comes over and hits a region which is complementary in shape and other properties, which we'll discuss in a minute, so that it sticks. It binds pretty strongly. And this is what triggers the further pulling of the virus inside the cell. But this surface has to be complementary to this surface to make it work. Let's go and look at a little cartoon version of this thing, just to make sure that everybody's understanding what's going on here, because the details matter critically. Okay, here's just a little cartoon illustration. This is supposed to be a smaller protein, which will bind to this larger protein. The larger protein is this kind of greenish area. And this comes down, and you can see this is actually this, but it's when it's binding to this other protein. And you see these little circles. You probably can't read them, but these are the groups on the surface of the protein that interact with complementary groups on the surface of the other protein. For example, this red dot here has a little line in it. That's supposed to be a group of the protein that has a negative charge. And when it comes down and fits in the geometrically complementary region, it sits right next to a group on the large protein that has a positive charge. That's what this blue dot is supposed to be. And this region comes down, this is oily, and this sits adjacent to other oily regions of this protein, and so on. So the chemical properties are complementary as well as the geometric properties. And it's these interactions which act like little magnets to pull this in and bind it to the protein. This has been likened to a couple of things. First of all, it's like a key fitting into a lock. If you think of the big protein as a lock and the smaller protein as a key, if the key has to have the right shape to even enter into the lock, let alone to turn it. Or it's also been likened to a puzzle with a missing piece. The missing piece has to have the right shape and fit snugly in order to bind into the puzzle. Here's another analogy. We talked about bacteriophage T4 and COVID, and both of these guys have to hijack cells in order to reproduce. And so you can think of them as thieves or burglars in the night, and they have to get into your house in order to do their dirty work. And some viruses use tools like this crowbar. We can think of bacteriophage T4 and its injection apparatus as a way to force entry into a cell. It doesn't mince any words. It just breaks through. Of course, it uses a machine that is a whole lot more sophisticated than this, but it manually breaks through the cell. On the other hand, COVID-19 is a virus that uses skeleton keys, like a burglar might have. It has keys that might fit a variety of locks, and it tries them out. And if luckily one of them works, then the burglar can get into the house and ransack it. Okay, so we've heard a lot about COVID-19 and viruses and whether COVID will evolve into something more dangerous or where did it come from and so on. So let's ask ourselves now, now that we have some basics of viruses and proteins and their structure and so on, how might these things evolve? What kind of mutations might they undergo? And this slide talks about DNA mutations, but this can also apply pretty closely for RNA as well, since COVID is an RNA virus. So let's look. It turns out that mutations can be a number of different kinds of events. And the common denominator is that the mutation changes the sequence of the genetic material of the virus. The first kind of mutation is called a substitution mutation, where one unit in the genetic material of the virus is switched for a different one. And this happens by accident when a copy of the virus is being made. The machinery that is copying, making a new copy, sometimes makes mistakes. You know, everything on this earth, you know, is not perfect. So sometimes it makes a mistake and puts the wrong nucleotide or the wrong unit in a position where it should have put another one. Nonetheless, it gets kept, and that's called a mutation. Another kind of mutation is called a deletion mutation. And as its name suggests, that means that in copying the virus, a mistake is made where one or more units is accidentally left out. Insertion is when one or more units is accidentally added where it shouldn't be. And we have some other things too. One is a gene duplication, where a piece of DNA or RNA genetic material that codes for a whole gene, which is the information needed to make a whole protein, it accidentally is doubled. It's kind of like I have a photocopier, you know, you copy a page and then you have two copies of the same page. And sometimes the whole genome gets copied twice, and that's a mutation as well. So you have a bunch of different kinds of mutations. And again, the common denominator is that the DNA sequence or the RNA sequence has changed. And since the sequence of the DNA is the genetic information, that means that the genetic information has changed. And what can be done with this stuff? Well, I'm going to illustrate this, I'm going to switch over systems to one that is more closely studied. And that hopefully will illustrate things, and we'll go back to the virus. But right now I want to talk about malaria. We've all read that malaria is transmitted by mosquitoes. Mosquitoes carry these little single-celled organisms called Plasmodium falciparum. We'll just call it malaria. And it gets into the bloodstream of people and it eats their red blood cells, and it uses that to make copies of itself. Now, it turns out that the humans have undergone some mutations over centuries and thousands of years. And some of the mutations that have spread in regions of the world where malaria is prevalent help a person resist getting a bad case of malaria. And perhaps the most famous mutation is the sickle cell mutation, the sickle cell gene. Everybody's heard of sickle cell anemia, and that's a terrible disease. But it turns out that when you have just one copy of the sickle cell gene, you're not really sick from that. What's more, it gives the person who is carrying that some resistance to malaria. And here's a little map of part of the world showing in light blue regions that have malaria, and in light red regions that have a lot of people that have the sickle cell mutation, and then in purple the place where they overlap. So you can see that the sickle cell gene occurs primarily in regions of the world where there's lots of malaria. And it's a good thing too, because it helps. It helps the person resist malaria. And that's an example that is oftentimes used of the power of Darwinian evolution. Here's evolution in humans giving these folks the great ability to resist this deadly disease. And it is a good example of Darwinian evolution. But let's take a little closer look at what exactly the mutation is and how it works. Okay, this, I don't want to scare you, but this is a drawing of the molecular structure of hemoglobin in some detail. And you might be able to see each of these little balls here. Those are the units, the amino acid units that make up the chains of hemoglobin. So this thing is just regular old hemoglobin that most people in the world have. And on the next slide, this is the actual amino acid sequence of the two separate genes, two separate chains of hemoglobin that appear in people. And each of the chains is over 140 units long. Everybody has the very same sequence. Starts with, say, here's an L and an S and a P, A, D, K, T, and they stand for particular kinds of amino acids. And their identity and sequence is what makes this hemoglobin. Well, what changes in sickle hemoglobin? What changes in the mutant form of hemoglobin? Well, it turns out that of those 140 plus 140, almost 300 amino acids, just one of the units has changed. One of them changes right here. So that particular mutation, doesn't change much at all. We already had the hemoglobin. There's just one crummy little unit change. And it turns out, though, that changes a particular spot. You see this yellow thing here and another one over here. This is the amino acid position that is switched. Everything else is exactly the same. But fortuitously, let's just look at this side here. Let's ignore this over here. Fortuitously, that makes a little bump on the surface of one hemoglobin that can fit in the depression of another. And because hemoglobin has twofold symmetry, this one has a bump that can fit into here, and this one has a bump that fits in here. And essentially, they can polymerize. They associate, they stick to each other. And that causes problems because when you have too much of it, the sickle hemoglobin congeals and forms fibers which make the red blood cell really stiff and damage it. And that's what causes the disease. So the point I want to make is that this well-known example of Darwinian evolution is the tiniest change, and it's helpful in the circumstances, but it's also gunking up the role of hemoglobin in the human body. Here's a slide showing all sorts of other mutations too. There's a handful of mutations that help with sickle cell disease. And the only point I want to make here is that most of the mutations, or pretty much all the mutations, break genes. That is, they take genes that were already there, and by breaking them or making them ineffective, somehow that gives some protection to malaria. A person is resistant to malaria. So this is what mutation does. That's my point. So let's go back and ask, well, what role does evolution play in describing viral diseases like COVID? Well, the first thing to know is that the origin of viruses is roughly as mysterious as the origin of life. Nobody knows how they arose. Here's a recent paper from 2019 saying viruses are ubiquitous parasites, but a consensus scenario for their ultimate origins is still lacking. And when you see that in a paper, it generally means that nobody has a clue how such a thing could have occurred. So one point I want to make is that nobody knows how viruses arose in the first place. Well, okay, how about coronaviruses? When did they begin, and did they pop up recently, and are they changing so that they will do great damage to people? And here's a relatively recent paper from 2013 saying that coronaviruses are very old. That is, the time to the most recent ancestor for coronaviruses is likely to be millions of years. And in the paper, they say it might be 300 million years ago that coronaviruses appeared. And they've had this roughly the same structure ever since. So the point is that these viruses, whatever they do, are not changing their general structure very much at all. So why do people worry about viruses mutating? Well, you remember the virus, the key interaction for viruses like coronaviruses is to have the spike protein be able to stick to a protein on the membrane of the organism it's invading. If it does that, then it can invade the cell and take over. But most proteins are not fit by the spike protein. You know, it's rare that you get this interaction. So what does mutation do? Well, let's go back to our little drawing of a smaller protein binding to a bigger protein. This is the same drawing that I showed you before, except that you see this red X means that this no longer can bind in here because I have a blue circle here, and this is now considered to be a positive charge. And if this bound down to here, it would be right next to a positive charge on the larger protein. And we know that like charges repel each other. So this would not be able to stick to this. This would, come right out again. It would not stick at all. But keep looking at that blue dot up here. But if a mutation comes along and changes that to a negatively charged amino acid residue, just that one change, like the change, say, in sickle hemoglobin, there was one crummy little amino acid change. If that occurs, now there you can have a negative charge next to the positive charge. And if you have other things that are complementary here, now this can stick to the bigger protein. And if this is a spike protein, this is a receptor on a cell, that allows the COVID-19 to come in and bind. So the point is that mutation and evolution are very important public health concerns of viruses. If you change this interface here, it might be able to bind to more cells or different types of cells, and that could make it worse or less severe for people. But it's not changing the basic shape of the virus or the basic properties of the virus. Where they came from pretty much remains a mystery. So let's see. I will quickly say, heck, I want to talk about my book since, since I have the opportunity. This fits very well with what I wrote in my book that was published last year, Darwin Devolves. And generally it says evolution is real. It's true that evolution is real and that mutations occur and sometimes they can help. But the interesting thing is that we have experienced another scientific revolution because new equipment has been invented to easily sequence DNA and discover the genetic information of organisms. And the long and the short of it is that we are able to track down helpful mutations in organisms. And the overwhelming number of helpful mutations, surprisingly, have turned out to be ones that break or degrade pre-existing genes. Functional coded element is just another phrase for a gene. And so like with the malaria mutations, other than the sickle mutation, they all break genes, and that helps a little bit. And so it's selected in an environment in which there is malaria, but now those people have lost genetic information, and they will continue to lose genetic information. So it's a lot easier just to break something and get a helpful effect than it is to try to make something like, say, bacteriophage T4 with all of that fancy molecular machinery. And so I write in the book that Darwin's mechanism of random mutation and selection is dominated by what I call poison pill mutations. That is positively selected helpful mutations that cause the loss of function of a gene. So Darwin's mechanism works pretty much by squandering genetic information for short-term gain, throwing something away because in the moment it's helpful, but then it's gone forever. I have some examples of this, but I think I won't talk about them right now so that you'll have a reason to go out and get the book. But here I think is a good example of a Darwinian evolution at work, or a good illustration of it. There were some criminals who were trying to get away from the army in a particular country, and they got ahead of them. And in order to escape, they blew up the bridge that the army had to cross to come and capture them. So from the point of view of the criminals, this was a helpful change. This was a beneficial mutation. And this is pretty much how Darwinian evolution works, that random mutations frequently break genes, and that in some circumstances, in some environments, that helps an organism survive longer. But it doesn't make anything, worth writing home about. It's not constructive, it's mostly destructive. Okay, so I don't know if any of you know this, but not everybody agrees with me on a lot of this stuff. And a few people have actually criticized my work, and I've responded to them at various places. First of all, at my university, at Lehigh University, my faculty web page, I have a number of most scholarly articles replying to critics. And at the website of the Discovery Institute, I have a number of web posts and essays and so on. But most recently at the website of my new book, darwindevolves.com, I've got a collection of responses to all of the critical reviews, and I show why everybody else is completely wrong and I'm correct. Okay, so thank you very much for listening. And Walter, I'd be happy to answer some questions.
Okay, thank you so much, Dr. Behe. If any of you have any questions in the chat window, whether you're in YouTube or you are in or you're in Zoom, please go ahead and type in your questions now, and Dr. Behe will attempt to answer those. Unfortunately, because there's so many people, we can't do this by voice, but please do feel free to put any questions you may have in the chat window of either YouTube or Zoom. So we have a first question. This is an interesting question, Dr. Behe. Is the coronavirus man-made?
As far as I can tell, there's no convincing evidence that it was. These days, for folks who don't follow this, these days, scientists have the ability to go in and change particular units, particular sequences of viruses. And it was suspected that perhaps a laboratory had altered a pre-existing coronavirus in a way such that it would be more deadly or more effective at invading human cells. A lot of people have looked into that, and it's up in the air. I don't have any extra information that other folks don't have. So it's bad enough as it is. So we may as well just think of it, I think, as a natural phenomenon.
Okay, next question, and I'll ask some questions that are more scientific questions, and then we'll get around to some of them about ID. Carl asked a question: what is involved in developing a vaccine?
In developing a vaccine, generally the scientists will take the virus or parts of the virus and cut it up or break it into pieces and try to inject it into some poor animal and have the animal produce antibodies, let its immune system try to develop antibodies against it. And if they find a fragment that elicits a very strong response to the from the animal to that, then they can try to use that in people. That is an inactive piece of a virus. And if you inject it into a person, your immune system kicks into gear and starts to develop immunity against that piece. The idea is that when now a live virus, one that would cause you real harm, comes to a person who has been immunized, your immune system has already been primed. It has all of the specific machinery already deployed to interact with that virus and destroy it before as soon as it appears. The problem is that our immune systems are terrific, but they take a little while to kick into gear. And once you get infected, it's a race between the virus trying to replicate in your cells, which usually trashes your cells and makes you very sick, and your immune system trying to develop these specific tools just to get that virus. It's a tricky job because your immune system has to recognize invaders like viruses and destroy them, but not cause harm to your own cells. So it has to be able to distinguish between you and the invader, and that's not easy for molecules which can't see. So that is a very very broad overview of vaccine development.
Okay, great. Thank you, Dr. B. Okay, here it comes, the design question.
Great.
Where does the design information come from for creating the organisms and their mutations?
Well, exactly where it comes from, as I've written in my book, is a question that's separate from whether a system was designed or not. Let me liken this to a favorite example of intelligent design, folks, Mount Rushmore. Okay, if you had never heard of Mount Rushmore and you suddenly came upon it, you might say, "Who? Look at that! That I can tell somebody designed that. That was made by an intelligent being somehow. I don't know what tools he may have used or whatever." If you ask the question, where did the design information come from to make Mount Rushmore? Well, what do you mean? You mean, did it come from some architect? Was it a space alien that landed and decided to make this? Or that's a much more difficult question to ask. But the great thing about intelligent design is that you can look directly at the system and you can tell just by how the system is put together whether it was designed or not. And that is, if it's a nice case like, say, Mount Rushmore, or say bacteriophage T4, or the bacterial flagellum, or other such things. So I'm going to pass on that. I'm a Christian. I certainly think that the designer was very likely God, but that is not directly, you can't conclude that directly from the scientific evidence. The scientific evidence doesn't take you that far.
Thank you, Dr. Behe. Can you clarify if the mutations are related to microevolution, or do you believe that microevolution can lead to macroevolution?
Well, the mutations of the virus are micro changes. And in my book, new book, Darwin Devolves, I pretty much show that that microevolution is the only thing that Darwinian evolution can do. In order to get macro changes macro molecular changes, say building a bacteriophage T4, or building a mammal, or anything else, then you need coordination and you need foresight. In other words, you need design. So the thing is, microevolution is real, but Darwinian macroevolution does not exist.
Dr. B. Another question from, from Jennifer. I've been told that viruses evolve in such a way as to not kill off their host and thus become less dangerous over time, at least the coronavirus. What are your thoughts? Now, I've heard things, in other words, like, for example, Ebola, well, it kills the host. So what good is a virus if it kills off everyone and then eventually it can't be replicated at all if it's killed off all the people and it kills them off quickly? So I think that's a great question.
Yeah, that's right. Yeah, people have investigated that, and I've read things on either side that, yeah, it will evolve to be milder, so it does not kill off the host, but it kind of reaches an agreement with the host to live there but not sicken it. And I've also read other things that says, no, it does not become less virulent so as to not kill its host. That as long as there's enough hosts around, it will be happy to kill them off without a thought, since viruses can't really think. But the most important thing for my purpose is that all of those scenarios presuppose the ability of the virus to change, to do such things. And I think they can, but even that would be microevolution. Even that would be changing the amino acid residues or a few other things on receptor proteins. So you can get medically important changes by mutation and selection, but they're all very small microevolutionary changes.
Okay, thank you. Okay, so Dr. B, this is from Jim. I guess some of his daughter and some friends changed it to 8 Girls Nikki for some reason. But anyway, this is what Jim asked. The virus causes great death, suffering, and destruction. How does this fit into intelligent design? I guess this is some type of problem of evil, potentially.
Sure, yeah. The problem of evil has been around for a long time. How can bad things happen to good people? But it doesn't really, that's a question for philosophers and theologians. Fortunately, I'm a biochemist, so I don't have to answer that question. What I do say, though, is that you can tell that machine guns are designed, that nuclear weapons are designed, all sorts of things are designed whose can have effects that would be hurtful. But this does not mean that the designer of viruses has it in for us. You have to look at a broader picture. Bacteria, which sometimes infect people and cause disease, have lots of good uses too. If there weren't any bacteria around, life would not exist, including us. Same thing with viruses. Viruses do have their uses. They can do some positive things. So you can't just look at this particular phenomenon right here and say, oh, that's bad. I will decide my worldview based on this. There's a lot of things going on in making life that can persist, and some of those things will have bad effects on particular people at particular times, but they are likely to be very good for life in general. So I think these are examples of those, that they might, just like if we come across a lion, it can pounce on us and eat us. Still, lions are nice to have around, and they probably do the world a whole lot more good than harm. In the same way, bacteria and viruses and other such things, they might cause us harm, but they might be very necessary for life on Earth.
Thank you, Dr. B. Great answer. I enjoyed that. I just would not want to be on the receiving end of that line. It's beautiful. Okay, we have time for a couple more questions. I think you'll like this one. Can you share the marked difference between Edge of Evolution versus Darwin Devolves? What were you trying to accomplish in Darwin Devolves that you didn't accomplish in Edge of Evolution?
Well, in Edge of Evolution, I was asking the question, what is the most that we could expect a Darwinian mechanism to be able to do? And so I looked at how many tries it took, say, malaria, to come up with resistance to the antibiotic chloroquine. And so the focus of that was, without any other consideration, how much can we expect a Darwinian mechanism to do? And even with it going full steam, it turns out, as I write in the book, that it can't really accomplish very much. With Darwin Devolves, I showed that it can't even accomplish what I thought it could accomplish in Edge of Evolution, because the evolutionary mechanism is actually strongly devolutionary. As I showed with some of the malarial cells or malarial mutations, it's very common for a helpful mutation to be one that breaks a gene. And so suppose that, the initial example I use in the book is a polar bear. Polar bears are supposed to be descended from grizzly bears over a couple 100,000 years. And these days, with the DNA sequencing revolution, scientists have sequenced the entire genome of the polar bear and of the grizzly bear multiple times and compared one to the other and asked the question, well, what changes in genes have occurred between the grizzly bear and the polar bear? And it turns out that the top 17 genes, most of them, 3/4 of them, are ones that break genes or degrade their activity. So it turns out you can make marginal changes, like between grizzly bears and polar bears, which actually can still mate with each other. But you can make those changes. You can fit a species to a particular environment, like the polar bear. But overwhelmingly, Darwinian processes, which of course can't see ahead, tend to break genes, and that helps. And the polar bear will never have those genes back. It's never going to become a bear that eats berries again, lives in temperate regions or any such thing. And get the book, because there's lots of other examples of that.
Okay, great. Thank you, Dr. B. Okay, here's another question, probably more not necessarily scientific, but maybe a more philosophical question, but we'll throw it your way anyway. Do you think that many criticize ID not because they don't agree with the arguments in favor of it, but because they cannot accept the theological implications?
I would have to be a mind reader. And there's a whole lot of people who don't accept ID, and they can do so for various reasons. And there certainly are people who don't like the theological implications of intelligent design, and they can certainly be resistant to it. There are other folks who think that, they're pretty sure that Darwinian processes can do the job anyway, and they tend to be folks who don't study the systems very closely. But I would hesitate to ascribe any particular motives to critics, but I'd prefer to focus on their arguments. And so far, I have not seen any arguments on this intensely controversial subject that have countered any of the points that I've made in my 3 books on the subject.
Okay, thanks, Dr. B. Here's a good question from Jordan. Are there any studies that have proven that in other words, novel, new genetic information can be produced? And I'm presuming that they're answering that question through Darwinian mechanisms. Have you seen that, or do you think there's any evidence for that?
Well, the short answer is no, there hasn't been. But when you get into a discussion about this with people who are supporters of Darwinian theory and so on, you have to say what you mean by information. For example, some people would look at folks with broken genes, like those who are resistant to malaria, and say, why, look, they have gotten more information because now they're resistant to malaria. So, and you can counter and say that, they used to have a functioning gene and now it's broken. And they might come back and say, well, la-di-da, still they have this different trait. So they're going to count that as new information. In my in Darwin Devolves, and a paper in a biology journal I published a while back, I try to nail down exactly what I mean by saying that Darwinian processes mostly break genes and throw away information. And I have to define exactly what I mean by information: a sequence of DNA that codes for a gene or codes for something else. And I've had a modicum of success, just a modicum of success trying to get folks on the other side to concentrate on the information, the coded information in the gene. But it turns out that I've been in this business a while, and communication between different folks is with different views is very difficult. It's very hard to get people to see or focus on exactly what I think is the problem.
Okay, we have time for one more question before we have to do a little housekeeping with our chapter here, Dr. Behe. Mm-hmm. And the question is from KJ Welker. Why do most people, and this is COVID related, so why do most people infected with COVID-19 demonstrate no symptoms while others die from an inflammatory response? Why the dramatic differences in responses?
Well, that's of course not known right now. Lots of people are working on such questions, but here's what I think is most likely, and that is your body and your lung tissue is a very complex, intricate environment. You saw the cryo-electron microscopy structure of bacteriophage T4. Crummy little bacteriophage has all of this intricate stuff. And so a virus like COVID, when it's going and trying to invade cells, it will interact with many proteins besides just the receptor protein. So maybe in a lot of people who are resistant, they might have a slightly different form of one protein or another protein or a third one that makes it harder for the virus to replicate or go through its entire life cycle. So a lot of other genes are likely to have an effect on the success of COVID in replicating in a human cell. So what people should do is sequence the genomes of folks who are resistant to the virus and who are susceptible. And I'm sure some folks are trying to do that, but their results have not yet been announced.
Okay, thank you, Dr. Behe. That's all the time we have for questions. And Dr. Behe, just so you know, there are like 60 plus additional questions waiting for you. So perhaps we can send those to you and you can select a few of them and respond to them, and we'll post, and we can post that at a later date, if that won't be too much of a burden on you.
That would be great.
Okay, well, thank you so much, Dr. B. I really enjoyed this. This was very educational for me, and I'm sure everyone else as well. I would like to turn it over now very quickly to the president of the SCN SoCal chapter, Jim Johansson. Jim, I think you worked out your issues here, your audio issues. Would you like to come out of mute and talk to us for a couple of minutes? Hello, Jim. If you're talking, we cannot hear you. You're still on mute. I will unmute you, Jim. Hello, Jim.
So I think that was an excellent talk. Thank you, Dr. Behe, for that wonderful information. I'm also very grateful for the discussion that we've had. And this is actually one of the reasons why we have the chapters. So I'm the president for the Southern California chapter of the Discovery Institute Science and Culture Network. And the mission of our chapter, and actually all of our chapters, is to advance the understanding that human beings and nature are the result of intelligent design rather than blind and undirected process. And our focus is in the Southern California area, but also since we have a very global audience, we welcome you and we want to embrace the dialogue that would go even beyond that area. And so we are a chapter of the Science and Culture Network, which is a project of the Discovery Institute's Center for Science and Culture. We're grateful for the Discovery Institute's sustained effort as a thought leader in intelligent design and for the support of our chapter, and also for great leaders like Dr. Behe. We also welcome the opportunity for the collaboration that we're doing with the Colorado and Houston chapters in this event, and this is something we plan to do in the future. And the global outreach that Discovery has is, it's interesting to see how that's going as we develop and we do further events like this. We also would like you to join in on our mission of engaging our communities and culture with the power of the ideas we are exploring now. And one of the ways you can do that, you can subscribe to our email list. We're planning for a number of events, both locally and more of these online virtual events. And I posted the information for how to do that. You can look at our webpage, and I'll give you that information briefly here. Remember, this event is free. And if what you find is something that you appreciate, of course, we would welcome your contributions. You can go to the website, https://socal@scienceandculture.network, or you can also just go directly to the Discovery Institute website. Let me introduce the very capable chapter leadership that we have. Walter Meyer is a vice president who has been doing the MC for tonight. We're very grateful for his efforts. Julie Smith is our secretary. Daniel Kerr is our treasurer. Joe Vaughn is our outreach coordinator, and myself, the president. One other thing I just wanted to mention, since we were talking about Dr. Behe's book, we would welcome to have a venue, a book club, if you will. We could go through Dr. Behe's book, Darwin, Devolves, or some other materials or other ways that you would like to engage with in this material. It could be another venue to address some of these questions that you might have. The chapter's mission is to go a little bit deeper and more local, but we also can be helping out in this global type of engagement that we're having here. So that's a quick little synopsis I just wanted to offer up, and thank you all for attending.
Okay, thank you, Jim. Julianne, did you want to talk for a few minutes to the folks before we close out?
Yes. Hi, everyone. Thank you so much, everyone, for joining us this evening. And one of the things I'm going to be doing is I'm going to be sending out a follow-up email. This is the first of these events, these online events that we have done. And we plan on doing several more of them and getting other speakers. And we would really welcome all of you back. This has been a great first event. I thank you, Dr. Behe. It was amazing. And I think we all, think so. So in any case, you're going to get an email from me or from us just in general. And we're going to just ask you a few questions about like where in the world you're from and how, did you hear about us through Facebook or was it from Evolution News and Views or whatever? And then we're also going to maybe ask you a few questions about speakers you might be interested in. So please stay tuned and just thank you, everyone. We hope to get another one of these events going, maybe even like a month from now. So stay tuned, everyone. All right. Thank you so much.
Thank you so much, Julie Smith, our secretary. Again, everyone, that will conclude our program. Thank you again, Dr. Behe, for joining us. We truly enjoyed it. Everyone, we will have this posted up on YouTube. There will be a recording, and we will also share that with you. Again, we hope to do this very soon in the near future. Thank you again, everyone who attended. And we will take these questions and we'll try to get Dr. Behe to answer at least some of them, because of course, he's very busy as a biochemist at Lehigh, but we will see if we can get a few of these questions answered and posted up on YouTube as well. Again, thank you everyone for joining us. Hey, we hope you will have a great morning, afternoon, or evening, wherever you are in the world. Thank you so much for joining us, and we'll see you again next time.